Okay, Bismillahirrahmanirrahim. Assalamu alaikum, ladies and gentlemen. Welcome back welcome to a new and the third and the last day of the international course on outbreak investigation and control of infectious diseases. Our first talk of today is by Dr. Sabah Farooq. The title of her talk is Tackling the Infectious Viruses, some of the examples from their work that they have conducted at their research institution. To introduce Dr. Sabah Farooq, Dr. Sabah Farooq is an assistant professor at Dr. Panjwani Center for Molecular Medicine and Drug Research at International Center for Chemical and Biological Sciences, University of Karachi. Dr. Sabah is associated with National Institute of Virology, Dr. Panjwani Center for Molecular Medicine, and is an assistant professor. She has a PhD degree in molecular medicine from the same institution and a postdoctorate in molecular and diagnostic virology from Mediagnost Diagnostics from Germany. And in addition to that, she also has her postdoctorate from the Department of Molecular Pathology from University of Tübingen, Germany. Recently, she has completed a comprehensive fellowship for the research and advanced training in virology and vaccine technologies in Indonesia by Comstech. She's working on antiviral discovery and is engaged in investigating molecular targets of the viral entry, replication, and pathogenesis. With this, I would thank Dr. Sabah for her time and for being with us. And over to you, Dr. Sabah.
Assalamu alaikum. Can you hear me, Hasima?
Yes, Dr. Sabah, we can.
Okay. First of all, I would like to thank you all, the organizer of Comstech and our respected guests from Infectious Institute of Iran for such an amazing and interesting training course on this important topic. I would like to share my presentation. Is this clear for everyone?
Yeah, we can see your slides. Can you please put it on slideshow?
Yeah.
Okay.
Okay. So I'm Sabah Farooq, and today I'm going to discuss briefly about some of the examples of our work which we have conducted on different infectious viruses. So starting with the virus, which is the causative agent of the recent pandemic COVID-19, anti-SARS-CoV-2 activity. So briefly discuss about the world after the pandemic. So the COVID-19 crisis aggravated the pre-existing weaknesses and proved to be a reality check for many aspects of the healthcare system. During the pandemic, the public health surveillance program and the available infrastructure were found to be not as consistent as optimal, and the health setup appeared to be unable to absorb or manage this type of sudden pressure and workload, especially in the system for the setup of acute care. However, this pandemic also created a once-in-a-generation chance for the transformational health system change. So the pandemic has mobilized and united many elements in terms of the drug discovery, drug development, process to seek the therapeutics against the SARS-CoV-2, in terms of like drug discovery and development or the rapid vaccine development. So what is the painful lesson we learned from the COVID-19 pandemic is the urgent need of the broad spectrum antivirals, which should be the host-directed antiviral, in order to prevent the viral pandemics of any unknown or known origin, and the ideal drug to limit the rapid spread of the virus to be the broad spectrum agent with the activity against a wide range of different types of viruses. So far, the available treatment options against SARS-CoV-2 include some repurposed drugs and others, which include remdesivir, hydroxychloroquine, azithromycin, some retroviral combinations such as Clatra and Paxlovid. But despite all such effort, there is currently no successful or broad-spectrum antiviral countermeasures to combat the potential SARS-CoV-2 or the potential against the SARS-CoV-2 pandemic. So we discussed different things. This work is done under the supervision of Professor Dr. Mohammad Iqbal Chaudhry, and we have discussed different options on the therapeutic strategies, either to do the random screening or the selected one. And the one option is to screen the existing broad spectrum antiviral, or to choose the molecule or the libraries of molecules or the databases to screen the molecules that have already potential against the therapeutic effect of SARS-CoV-2 and other viruses. In this way, we should develop the drug which is based on the genomic information as well as the pathological characteristics of SARS-CoV-2. So what we basically select the PCR positive SARS-CoV-2 sample and propagate it into the cell line. And after observing the cytopathic effect, we confirm this cytopathic effect by using the RT-PCR, and then check the infectivity of the virus, and optimize the plaque reduction assay for the screening of number of libraries of compounds. We have also confirmed the positive control by using the immunocytochemistry or the using the DAPI, fluorescence microscopy, the remdesivir, and simultaneously, our group is also involved in the isolation of different plants extract. So I collect those plant extract and screen against SARS-CoV-2, and also check the activity on those compounds or extracts are of preventive nature or of therapeutic nature, or either they inhibit the entry of virus or any other aspect. So what are the findings? Basically, initially we check the propagation and cytopathic effect into the vero cell line ATCC CCL81, and we observe on daily basis like 24, 48, and 72 hours. After confirming the cytopathic effect, we confirm it by RT-PCR, and we passage these cells up to passage number 6 and confirm the availability of the virus into the vero cell. After this confirmation, this image shows the control. These are the control cells with only the control vero cells fully propagated and grown in a normal morphology. And this, the next one is with the remdesivir, normal morphology, normal growth. But when we see the vero cells with the SARS-CoV-2, marked reduction on the number of the cells were observed along with the altered morphology. And we check the remdesivir, the positive control on the infected vero cells, and we observe the growth was retained and with the normal morphology. So what we did, we basically screened large number of medicinal herbal plant extracts such as clostridium, mentha, pipetta, and others. We have also screened repurposed drugs such as antihistamine, antimalarial, etc., and the libraries of natural and synthetic compounds which include terpenes, alkaloids, and commercially available natural products, etc. So what we have been able to achieve so far, these are some glints, and I will proceed with this. This is the US patent, published US patent on the prevention and therapeutic effect of SARS-CoV-2 by using the mentha, pipetta, and clostridium mentha. So we have evaluated these are different medicinal plants with the % reduction against SARS-CoV-2 and moderate to a significant % reduction. So we have selected some plants on the basis of their potential against SARS-CoV-2. We performed the cytotoxic evaluation, and we confirmed that the cytotoxic, the CF, which is clostridium mentha and mentha pipetta, were observed as non-cytotoxic. So we proceed for the plaque reduction assay for the evaluation of the dead zone. And over here, we observe the marked reduction in the plaque formation, even in the crude extract as well as in the ethyl acetate fraction. Sorry. These are the % reduction, and over here we observe that the clostridium mentha and mentha pipetta ethyl acetate fraction exert significant marked reduction up to 90% against SARS-CoV-2. We have also checked the treatment point assessment, like these extract, crude extract or the fraction either exert their effect on the preventive mode of action or inhibit the entry or the therapeutic. So over here, these plots shows the prophylactic and entry point. And over here, we see that the hexane extract of the clostridium markedly reduced the percent viral copy number, followed by other ethyl acetate and butanolic extract. As far as MP, which is mentha piprata, is concerned, the ethyl acetate fraction of mentha piprata reduced percent viral copy number up to 0.3, which is much lesser than the positive control chloroquine phosphate and remdesivir. As far as entry point is concerned, again, FC hexane exerts significant reduction, and the an MP, ethyl acetate MP, exerts a significant reduction. As far as therapeutic is concerned, the FC hexane was not produced as such significant reduction as compared to the crude extract, while the EA of the mentha piprata still produces significant reduction of the percent viral copy number, indicating its therapeutic effect as well. So, if you are talking about the clostridium crude extract, it produces its effect on the entry point, followed by the therapeutic and the preventive mode of action. While MP, which is the mentha piprata crude extract, produces its effect on the entry, followed by preventive and similar to the therapeutic effect as well. If we are talking about the fractions, not the crude one, the fractions, ethyl acetate fraction was observed as the significant one, followed by the hexane and the butanolic extract. This butanolic extract is only for the clostridium, not the mentha piprata. So after observing these results and after seeing that these clostridium and mentha piprata crude extract and fraction produces their effect in reducing the SARS-CoV-2 potential, so we move towards the subfractionation at the concentration of 10 microgram and even at the 1 microgram per ml. So we check the prophylactic effect on the 10 and 1 microgram per ml, and over here, and we selected the clostridium for the further subfractionation. And over here, we observe that the F20W2 produces the marked reduction in percent viral copy number at 10 microgram per ml and 1 microgram per ml as well in the prophylactic, indicating the preventive effect of this subtraction. If we are talking about the entry point, the F20W2 produces significant effect at the entry point at the 10 microgram per ml, but did not produce its effect or the significant effect at 1 microgram per ml. As far as therapeutic effects are concerned, at 10 microgram and in 1 microgram, this F20F3 produces significant effect, indicating its therapeutic potential as well. So we selected this F20W2 subfraction. It produces its effect, preventive and therapeutic effect, even at 10 and 1 microgram per ml, and it also inhibits the entry of the virus at 10 microgram per ml. So the conclusion of this first study is that the subfraction of clostridium against SARS-CoV-2 produces its effect even at 1 microgram per ml, indicating its preventive and therapeutic effect against COVID-19. And this results may be further lead to the development of any antiviral formulation, and the possible mechanism of action will be identified in future studies. So now moving towards another study, which is based on the identification of the resistance-associated substitution in hepatitis C virus and their impact on the direct-acting antivirus. As we all know that the hepatitis C virus is the chronic severe disease, and around 58 million people are chronically infected with HCV. And a large number of infected individuals remain unaware of the HCV infection until the development of the serious complications, which includes the liver cirrhosis and hepatocellular carcinoma. So this hepatitis C virus, this is the normal liver, and it converts into the acute hepatitis. This acute hepatitis, the normal in 0 to 6 months, it is cleared by the HCV clearance, but 70 to 80% lead to the chronic hepatitis, and among these 70 to 80% in the during time frame of 10 to 40 years, 15 to 20% lead to the liver cirrhosis, and 75 to 85% start progression and formation of the fibrosis. And among this liver cirrhosis, around 3 to 4% converted into the liver cancer. So these are the prevalence and the distribution of the genotype worldwide. And over here we can see in the Pakistan, the genotype 3 is more prevalent, followed by the genotype 1 and genotype 2. If we are talking about the epidemiology of HCV in Pakistan, the most prevalent serotype is genotype 3, and the most predominant provinces of this HCV are Sindh and Khyber Pakhtunkhwa, followed by Punjab and Balochistan, and some region of FATA as well. So why we are focusing with this HCV genotyping? Basically, this HCV genotyping is the strong prognostic marker for the HCV antiviral treatment. These are the timeline for the HCV antiviral treatment, starting from the interferon therapy and the combinational therapy, then the approval of the protease inhibitors, NS5B, NS3A protease inhibitor as well. And these are the direct acting antivirals, some protease helicase inhibitors, Previr, and NS3-4A protease inhibitors, some are NS5A inhibitor, and some are RNA dependent RNA polymerase inhibitor, which are NS5B polymerase inhibitors. And these are NS5B, NS5A inhibitor, and their combination and their duration and the cost of HCV treatment in Pakistan. So as we are working on the resistance associated mutation, these mutations are basically any amino acid change in the consensus sequence that associate with the reduced susceptibility of virus to one or more antivirus. So if the resistance occurs in the NS region, it leads to the resistance against the direct acting antivirus. So what are the findings? Basically, we collected samples from different districts of Karachi and different cities of Sindh as well, which include Hyderabad, Larkana, and different cities. We basically performed the amplification of NS3 and NS5B region, followed by the Sanger sequencing. For the genotyping, we selected the 5' untranslated region, which is very beneficial for the identification of the genotyping. And then we construct or analyze the RAS analysis, and then phylogenetic analysis, computational analysis were performed by all these vessels and Viper, etc. So this is the demographic data from our samples, and we observed that 54 female patients were infected and 46% male patients were infected with HCV. If we are talking about the age-gender association or the average age, we observed that the average female age was 45 and the median age was 45.61 and the median was 45. And the average male age was 43.69 and the median age was 45. Distribution of HCV in age group, we have observed in our samples, the age group of 41 to 50 was most infected, followed by 51 to 60 and 31 to 40. And this is the correlation between the age groups, and we observed that the median viral load was slightly higher in elderly patients as compared to the younger patients. So moving towards the NS3 resistance associated substitution analysis. And this is the list of the mutations we have observed in our samples of NS3. And this is the prevalence and the frequency of mutation. And over here, we have observed that Q41 into H and Q80 into K and E. And these are the position of mutation we have observed in our samples. And we have also checked the prevalence of NS3 RAS genotype 3A worldwide as well. And this green color indicates the samples from NIV. And we have observed Q41 into H, 0.032%, Q80 into K, 0.032, and Q168 into our asparagine, 0.096%. this mutation Q168R lead to the resistance against the glycoprevir. So what is the impact of these mutations on the antivirals? So the mutations we identified in our sample linked to the resistance of different types of drugs like glycoprevir, tenofovir, and favipiravir. Sorry, A156 to T associated with the resistance of semiprevir, Q80 into R is resistance to the semiprevir, Q168 associated with the resistance of semiprevir and favipiravir, and D168R is associated with the resistance of the glycoprevir. So most of the resistance we have identified during our studies are associated with the resistance to some certains of the antivirals which are already available. And when we go towards the NS5B RAS analysis, this is the list of NS5B, non-structured protein 5B mutations in our samples. And over here, we observe these two important mutations. If we are comparing NS3 and NS5, we observe more mutation in NS3B region as compared to NS5. And over here, 16 when replaced with 316 position with asparagine, we observe that 0.05% frequency of mutation. And this is the comparison with worldwide. And over here, we observe that this mutation, which lead to the resistance against sofosbuvir and dacibuvir, is 0.06%. Phylogenetic analysis, we selected for the phylogenetic analysis of NS3B of around 31 samples, and these are the data from the worldwide data. And we observed this is the clade, or this is the NIV samples, the red one, which are submingled with the Asian population. Some are submingled with the Asian population, and some are producing the separate clade over here, and this one as well. This is the NS3 region. And for NS5B, green one, the 35 samples from NIV. And over here, we observe the distinct pattern, the green one in NIV samples, and some are submingled. So we have identified different mutations that are associated with the resistance to different types of antivirals, and we have also identified the mutations or the diversity in the samples which we selected for the phylogenetic analysis. We have also found out the efficacy of the direct acting antivirals. During this study, we have selected the data of 202 patients which are included in this study, and they received the sofosbuvir and dacibuvir and ribavirin by the Sindh Government Hepatitis Control Program, and the choice of treatment is based upon the availability of the drugs. And over here, we observe the combination of sofosbuvir and dacibuvir is 0.08%, and the other combination 0.06%. And we have observed the sustained virological response overall 0.9%. So outcome of this study is that the most prevalent HCV is most prevalent in females as compared to males. The reason may be due to the piercing, maybe due to the blood transfusion and other matters. And genotype 3A is the most common genotype in the region. NS3 mutations are higher as compared to NS5B mutation, and the higher rate observed higher rate of the treatment failure are 9%. And we have also observed the higher number of mutation in C316N, 316-2N, and L159 to phenylalanine. And also our study shows that boxyprevir could be the highly effective drug for the NS3 treatment because of no resistance observed against this drug. And the phylogenetic analysis observed that the virus is highly diverse in this region, highly diverse in Pakistan as compared to other regions. We have also observed that the associated with the NS5, we provide the idea for the explanation for the drug of choice. And the present study indicates that the high rate of treatment failure, which I already mentioned, 9% in the HCV-RN. This study is recently published in scientific report. Now I'm moving towards and briefly explain one of our study which is based on the pathogenicity and the genomic diversity of the most circulating strain of the dengue virus in Karachi, Pakistan. During 2022, because of the flood in the rainfall, we have observed the highest rate of the cases in the dengue cases, and the most severely infected province of the dengue was province Sindh. And these are some samples collected from different hospitals or different districts of Karachi. Altogether, we have collected during the outbreak of dengue around 600 samples, and we have checked these samples by using the two primer sets, E and NS1 region, and we have observed 412 samples were found to be highly active, highly positive, with a 68.66% positivity rate. Then we checked these samples by using the serotype specific primers, and we have observed the prevalence of the most circulating serotype is the DNV1, dengue serotype 1, which is 84.70%, followed by 2 and 3. We have also observed some samples with the co-infection of dengue serotype 1 and 2. And this is the ratio of cases on the male and female gender. And we have observed 57.76% infected male as compared to 42.323% female. And this is on the basis of the age. And this is the average platelet count of each serotype. We have observed a reduction in the serotype of platelet count of the serotype 1, followed by 3, and as compared to serotype 2. Previously in Pakistan, serotype 2 was most prevalent as compared to serotype 1. But in our study, in our samples, we have confirmed that the serotype 1 is more prevalent as compared to serotype 2. We are working on the phylogenetic analysis and the sequencing of these samples as well. So after this, we have also checked the replicative fitness of the dengue serotype 1 and the dengue serotype 3 in different cell lines. We have selected cell line 0 and 27.5. And we have observed that the most drastic effects were observed in 27.5 as compared to 0 CCL81. Drastic effects were also observed in 0 cell line, but more rapid and more frequent were in the 27.5 as compared to 0 CCL81. We are also focusing on further replicative fitness investigation of these cultured 0 cells, and we will surely discuss in later sometime. So, in conclusion of this study, the results indicate that the most prevalent serotype in 2022 was the DENV1 with 84.61% of the cases, followed by DENV2 and DENV3. And the co-infection was also 3.12% was observed. The E and NS1 region of both shows the greater diversity as compared to DENV2, and the average platelet counts were found to be very low as 20,000 in DENV1 and DENV3 and co-infection as compared to the DENV DENV2. So, in the end, I would like to thank my supervisor, Professor Dr. Muhammad Iqbal Chaudhary, Dr. Samar, and Dr. Saira Banu for the extraction, Dr. Ishtiaq and Dr. Irfan for the genomic and bioinformatics study, Dr. Hana Iqbal, Ms. Zoha Khan, Mr. Sarmas Best, and Ms. Faria Khan. Thank you very much.
Thank you very much, Sabah. This is Kihan. Thank you
Professor
for,
Kihan.
and do you use the normal Vero or for SARS-CoV-2 culture, or do you use Vero E6?
Sorry, can you please repeat this?
Did you use Vero E6 or Vero normal Vero for
Normal
culture?
Vero cell line, it's ADCC CCL81, not the E6.
And what was the genotype of your COVID SARS-CoV-2 viruses?
Like this is the Wuhan strain.
Uh-huh. Okay. Okay, thank you. Any question?
Yes,
I have one question. Did you check the biocompatibility of your extracts with the normal cell lines? The extracts that show best activity. Did you check the biocompatibility of that extract with the normal cell lines? Is the extract biocompatible?
I
The
cannot
question-
hear her.
Which microphone is working now? Okay, so one of the audience asked, did you check the biocompatibility of the compounds you found in normal cell line? I think she means that wasn't it toxic for the cells at that concentration?
Yes, we have evaluated the cell lines, the compounds on the normal cell line as well. Initially, we screened the compounds on normal cell lines. And after getting the non-cytotoxic effect, we only select those compounds at that particular concentration. And then we use for the antiviral activity.
Okay, thank you. Any other question? Okay, thank you very much, Sabah.
Thank you.
Thank you
Thank you
for
very much.
joining us. And I hope you have a good time in Karachi and meet you in a couple of days.
Yeah, inshallah.
Inshallah.
Thank you very much.
Thank you. So, can you please give it? I have my slides here. Oh. Okay. This? Yes. Risk assessment and communication. Okay, thank you very much. Bismillah ar-Rahman ar-Rahim. Good morning, everybody. So we have a little change in the program because I didn't finish my talk yesterday. And I was told not to move away from this microphone, so I have to stay here today. So yesterday we talked about the risk assessment and communication. It started from this. We went through what Fauci said. Then we talked about that the risk is a function of the severity and probability of the hazard to happen. And then risk assessment, the process where you identify hazards, analyze it, and evaluate them, and determine the appropriate way to eliminate, mitigate, or control the hazard. So I think we stopped here. And well, the relevance of this subject to our talk or our course is that we have to decide whether we want to interact. I mean, to do something for an outbreak or not, and to what extent. So the objective of risk assessment is, first of all, to do a risk analysis to see the nature and level of each risk in terms of its likelihood and impact. So by definition, we want to see how big the risk is. So we have to know the hazards and the probability. And then to evaluate it, whether the risk is acceptable or not. I gave you this example of a common disease like common cold, which infects many people, but the consequences are low, so they're usually acceptable. And a disease like, for example, SARS-CoV-2 or COVID-19, which infects a lot of people, but the outcomes are fatal in a minority of the patients. So overall, the consequences are not so acceptable. And there are other examples for that. Then what you can do to control the risk. You have to develop some recommendations about risk or any treatment or any other kind of measure that can control or decrease the risk, or at least prioritize which group or which patients should be taken care of more rapidly. So these are the steps. And why do we need to do that in the public health setting? Because whatever we do is to reduce morbidity and mortality in the population. And well, our resources are limited, so we have to prioritize on which subject we want to spend more energy, more resources. Finally, we should have some vision on what will happen in the future if we take this action, if we take the other action, and see if the population accepts the consequences or not, or accordingly modify the recommendations. In fact, this is a balance between risk and benefits. So we must just make it just and justify whatever what we do. For assessing public risk and public health, some standard steps have been suggested by WHO. This is from a guideline on risk assessment for foodborne diseases, if I'm not mistaken. But the guideline could be applied on other diseases too. So first of all, we need a systematic process to make evidence-based decision, and then assess a combination of likelihood that an adverse outcome will occur following to the hazard and the possible consequences associated with it. We should have an estimate of that. We should see the current situation in the future. So what if the prevalence is increased? What if some other age groups are involved, and so on? The methods that are used for risk assessment could be qualitative or quantitative. Well, to my best of my knowledge, this is not really a mathematical field. I mean, nothing is very much clear. So it is a case-by-case basis. So we decide what to do. And it is very important that we have to communicate our finding with the stakeholders, both with the technical stakeholders who want to know our technical advice, and also the community who we want to help them and who we want them to follow our guidelines. And so at the end of this session, I will talk about risk communication with the public. Well, if you consider like a matrix of likelihood and consequences, and you see the likelihood would be certain, very unlikely to certain, and the consequences can be from minimal to severe. Our best chance, our target, is to have something here, some hazard which is very unlikely, and also the consequences are minimal. Okay, but usually the hazards, well, sometimes you can change the hazard, sometimes they are changed, but usually hazard is the characteristic of that problem itself. What you do is to change the likelihood of it to happen. So we see what we do. So if we find out that something is in the green zone, something here, it means that if we are talking about monitoring a disease, if it is in the green zone, it means that your current activity is okay. So you do not need to do any extra measure on that. Just routine procedure. If it is yellow, the risk level is moderate. So you have to focus on maybe your routine procedure is not sufficient. You must specify the management responsibilities, and the specific monitoring or procedure might be required. When it goes to orange, the risk is high, so maybe local capacities are not enough. You need to have help from the, I don't know, in your case, federal, or in our case, from central assistance to manage, to control the disease or situation. And the worst case is in the red region. So in the red region, you see that it could be either due to severe hazard, which is either almost a certainty or high risk, or major hazard, but again, almost certain. So if the probability is high, the consequences, it doesn't mean that both of them should be very at the topmost level, but the outcome must be, the combination of these two must be high. And in those cases, you are in the red zone. To give you an example, for example, measles is a disease. How do you analyze measles? What do you think about measles? Is it a very severe disease or not? Well, is it, what is the mortality rate or case fatality rate of it? Okay, I don't know. I don't remember. But anyway, I think that it is severe, but it's not very severe. It's not CFR. What is CFR, Dr. Eshrati? Do you know for measles?
Of course, yeah. The mortality rate depends on the setting of the hospitalization.
Okay.
As a matter of fact, maybe high, maybe low, but the consequences of measles is high.
Okay. So what consequences is either death or not? Okay. Anyway, finally, the morbidity is high. And what is the probability of getting measles? Is it high or not? Well, is it always high? Why not? Okay, in unvaccinated cases. So in this case, by vaccination, you can decrease the probability. Okay, or for example, with better treatment setting hospitals, you can take care of the consequences, reduce the consequences. And what about SARS? The original SARS. It is very severe, but it's very unlikely to see a patient like this. So anyway, it is in the red or orange zone, yeah. But what you are going to do is to reduce the risk. So what do you do with SARS? The probability is low, so you cannot do anything about the probability. So you focus on the consequences by enhancing the treatment facilities, for example. Well, precautions, maybe, but anyway, there's no, the probability is almost zero, so you cannot do anything to improve it. The goal is to move any of these to a lower risk zone. So for measles, for example, by vaccination, you make the consequences, you make its occurrence very unlikely. Okay, or for SARS, you cannot change the probability, but by improving the treatment setting, you decrease the consequences. So again, you move it from a orange zone to a green or at least red zone, yellow zone. Well, what you do in practice is that first of all, you have to form a team and ask the question: what is the risk that you are talking about? What are the factors that impacts it? You collect information, and then you do a rapid assessment. There are some questions. Well, there are different tools for this. I think yesterday, I assume Dr. Arashkia said about the risk assessment tools in the lab. So basically, the concept is the same. In the lab, something may happen. If you work with a dangerous pathogen, if it is released, it's very dangerous. So you try to decrease the probabilities by, for example, putting on the PPEs, by doing doing the experiment in a high biosecurity lab. Okay, and that was a BioRAM tool, for example. That's this. But this guideline gives also some question that you can answer, and then you discuss what is the perception of public about this risk. And finally, you do your recommendation, as I said, both to the authorities who should do the measures or take the measures, and also to the public who should accept the measures. And this process is reviewed and reviewed several times. So that was why Professor Fauci changed his recommendations several times during the SARS-CoV-2 outbreak. Well, when you assess a risk, you have many different components. First of all, you have to assess the hazard itself. Is it known or unknown? If it is unknown, so you must go have an unknown outbreak. You have to prioritize which one is more dangerous for you. So based on that, you may develop a new scenario. And what is the exposure? What is the exposure status of people, number of people likely to have been exposed, or people who are susceptible? You estimate them, then you check the context. Can you control the disease? Can your system work in this situation? For example, in West Africa during Ebola, one big problem was the shutdown of the health system itself in some of these countries. So when you do not have any health system, you cannot offer anything. And other factors related to the health system. So if you have a surveillance system, if you have diagnostic tools, treatments, etc. And finally, you document all this evidence. So this is one example of what you do. It's some type of an algorithm. Well, if you come from here to the end, there are several questions that you may answer. And for the lowest risk, you ask this question: is further exposure likely to take place, and is the population susceptible to disease? So if you have one isolated case and you do not expect it to expand, the risk is different from when you think that this may expand to the community. If the answer is no, then are a significant number of people currently affected? Well, if you have, for example, 1,000 people, but you do not expect to be 2,000, that's okay. By some way, they are isolated, so no. So the level of risk is low. What you do, normal procedure, is sufficient and meets needs of events and business as usual. So this is a low risk situation, and based on this perception, you do whatever you need to do. On the other hand, on the other extreme, when the answer to the first question is yes, is the disease severity moderate to high? Okay, if it is no, then again it goes back to this way and finally ends in low risk or moderate risk. But if it is yes, then would a high number of cases be expected in the future? Okay, if it is yes, is the disease severity high? So the previous question was moderate to high. Now, specifically if it is high, then are cases likely to be to present to healthcare in quick succession? This is very important for the health system because you do not want to overload the health system, do not want to overwhelm them. And this is, in fact, what we did during one of our goals during control of COVID-19, because we did not want to have all the patients at the same time in the hospital, so the hospitals are disabled. So by many means, by lockdowns, by, I don't know, even by vaccination, we try to flatten the peaks. So at each point of time, less people are admitted to the hospitals. Anyway, so if the answer to this question is also yes, are current capacity and control measures in place? Well, if yes, it is high risk. If it's no, it is very high risk. So what you do in the very high risk case, local capacity overwhelmed, requiring highest level of government to assist, perhaps international help, inform top level of management, activate emergency operations centers, consider high consequence control measures. Example, containment. So you see, based on similar or based on these or similar approaches, you can decide what to do in this case. And of course, this situation may change. When a new variant of a disease comes, so all the answer of some of these questions may change. So you have to assess again. And I want to give you a very, very nice example that I noticed during my stay in Pakistan. Well, I was in Karachi, and I don't know, have you seen the driving pattern in Karachi, and have you compared it with Islamabad or not? But I show you. So my question was, what is the rate of traffic accidents in these two cities? And I mean, is it related to this? To what happened? Okay.
Bring your business online with GoDaddy. Get all the help and tools you need to succeed. Join the millions of small businesses that trust GoDaddy.
Okay. Just listen to the noise. Okay, how do you describe the driving? What? Okay, so if that's normal, what is this? Okay, wide streets, were almost empty, driving very safely, no sound of horn, no noise, yes. What? Okay, that's a good point. That's a good point. Thank you very much.
For 1/5 of the world.
For what? I like it, yeah. But the question is that is the problem of car accident different in Karachi and Pakistan? Okay, so what is the risk what risk is being avoided during driving? Accident. Okay. How is the perception of risk in Karachi? Too much?
Even when you cross the road, you in doubt, oh my God, in front your guard, something is.
Okay.
This
What about Islamabad? Do you care what, I mean, are you worried about the car accident in this setting? Okay. And how do people manage the risk? So they have different perception of the risk based on her personal experience from Karachi, where she lives.
all even like from one side to another, take any other ride, pay an extra money, but please
Okay.
just receive.
So I mean that this is a very, very simple example of how do you perceive the risk and how do you manage it. Okay, and but how do people in Karachi manage the risk of accident? You know, they have this expectation that at any moment the car in front of you may change its line, so you must be very careful, and that's why they honk their horn so frequently. Okay, and you do not see this in Islamabad. So the measure that they take is different from the measure that they take in Islamabad, and the perception of risk, because the perception of risk is different. And what is the effect? I checked the car accident data from Pakistan, and I noticed that in, for example, 2021, this is the reported cases, vehicle involved in Islamabad, and this is vehicle involved in Sindh in general. So which one is higher? In Sindh. Okay. But if you normalize it to the people, to the number of cars, I didn't find the number of cars in these two cities, but I found the population of these two cities, or in two states, in fact. And you see the population in ICT is maximum 2 million in 2017, and in Sindh, 47 million. So if you normalize this data, you will see that the number of car, I mean, the rate of car accidents in Karachi is actually lower than Islamabad. So what does it mean? The risk management measures in Islamabad is effective in Karachi, sorry. So by doing that, they actually prevent car accident. You may argue this conclusion because you may say that the cars in Islamabad are newer, so they're more worried about, I mean, they report more frequently if they have any minor accident also. In Karachi, I saw a lot of cars which were hit already, so maybe they do not go to report the accident, or maybe many other reasons anyway. You can attribute it to many things. But at the first glance, I think that this risk management measure, which is deriving like this in Karachi, is really working. My point is that when you do a risk assessment and you want to do the risk management, it doesn't mean that you should have the best quality drug, you should not have the best quality hospital to lower the risk. Maybe by some of these kind of measures, you can also control it. Maybe by limiting access to some area, maybe by just asking people to wash their hands if they listen to you. So consider this risk management in your everyday job, and that's what you do. Just consider it how to do it in health business. Well, so what actually we did for COVID-19 control in the first early few months was mostly based on this kind of risk assessment. When we found our first case in well, we started in January 31 almost, the control of passengers from China. So based on this, at that time, the main risk was people who were coming from China. Then we found, as we established our lab, we found our first case in February 20. And well, some countries, you know, the first epicenter was a city in city of Qom in Iran. And we were asked about our opinion if we need a lockdown around Qom or not. And my personal perception and analysis was that we do not want it. So we advised against lockdown because on the same day or the next day, we found that some cases were tested positive from other cities of Iran. So the outbreak was already expanded to other cities. So there was no benefit in locking down this specific area. And also, I showed you a picture of Sana when she went to Qom as a field investigation team in epidemiology. And when they came back a week later, their estimate was something about, right now we have more than 30,000 infected people in Qom. So with so many cases, there is no need for lockdown. There is no benefit for that, in fact. So we were against it. Then we stopped the competition in early February, not early, just a few days after finding the first cases, the sports competitions, then national campaign for COVID-19 screening in March, early March. We expanded, of course, the laboratory for that, then the closure of subways, then the partial closures of the city, and so on and so forth. And for example, in the April, we tried to strengthen early case finding. So we changed the target group for testing the disease from hospitals to other groups. So based on this risk assessment at each step, you would decide and you would advise to do specific measures. Well, when you find the risk, when you analyze it, then you have to communicate it. to whom should you communicate it? To all stakeholders in general, but in their own language. If you're going to talk to the Ministry of Health, you should write a technical note, which if we have time, Arash will talk about documentation in that case. But when you want to talk to the public, you should use their own language. If you have to talk to the clerics, like religious cleric, you should use their own language. It was very brave and very difficult in Iran to lock down Imam Reza Shrine in Mashhad, for example. And I think in its history, it has never been as empty as it was during COVID-19, but they did it. And one important thing is you always should be as convincing as possible. So you do not force them, you must convince them that you should do this. And for that, you should be evidence-based. And then you give your advices. And if you want to publish it, to publish the result, it is like many other scientific reports. So you have the same structure, but in the description of the outbreak, you should clearly address the question, those famous WH questions of epidemiology: who, when, why, where, sorry, not why, and even if you can, why, yeah. And you clearly describe the measures that you have taken, or you advise based on what evidence, etc., etc. And sometimes you need to report to media. If you come back to this slide, the core concept here was coordination. So you have this media here and social mobilization somewhere here. Okay, here, social practice. So this is very important. And because why do you need to talk to people about that? Because they have right to be informed, and the authorities and also public should be informed to make an informed decision. You should build trust. During an outbreak context, it's critical to for the people to trust you. So otherwise, they will not follow your advice. And you want people to be a part of the solution. And you see, these are two examples of two big placards in two occasions. This is for chikungunya in Mauritius, and this is for avian flu in Turkey. And which one is better? Why? That's right. It is simpler. It is more clear. You know, you have very few messages in that placard, so you can easily grasp it. This one is very complicated. So my point is that, you know, advertisement is not is a skill, is a specialty for itself, and you may not be the right guy to prepare some kind of this advertisement. So you should have people for the social mobilization, for the advertisement. Even WHO sometimes asks for its GORN activities, sends requests to GORN members and requests for people for social mobilization. So these are people who can speak the native language of those people and also are very expert in social sciences. So it is not just messages, leaflets, posters, T-shirts, TV, and whatever. It is more than this. It is they must trust you. They must accept your advices. And people then feel that the risk is voluntary, is familiar. They have some analogy to some known risk with that. It is controllable, but somehow, especially if it is controlled by self, it is fair. So it does not affect only poor people, not rich people. Well, and it's not sequestrated to specific parts of the community, and it's not fair. Usually, these people have better understanding and better cooperation in these cases rather than these cases. And it is very important. It's a bit tricky, in fact, interaction, because if people do not trust you, they even try to kill you. And these are some examples. WHO and UN in general has its own guideline for traveling to dangerous places. So they give you training on how to deal with people, and you must be very careful. You must not, you cannot go to people who want to, for example, bury their dead people according to, for example, Islam, and then tell them that you should not wash before burying them. But you must convince them in some way, or you need a fatwa in this case, for example. Well, and the last thing is the infodemic. Infodemic is a big problem, especially it was in the case of COVID-19. And I again suggest you to watch that movie that I introduced you in the beginning of the previous talk. So even in that, you see that how infodemic is spread and how some people benefit from it. So they try for their own interest, they try to mislead people. And these are the characteristics of this problem. This is too much information, which are either false or misleading. If they are not false, they are at least misleading. And one big problem is that if they are, if they circulate around very frequently, they cause mistrust in the health authorities. So you cannot give them the right suggestion to do. They do not trust you. They do not follow your advices. And it can intensify or lessen outbreaks. So it's not just a matter of choice by someone. It damages all people. It's not just a personal damage. And for facing it, you must listen to the community concern. Well, people may say that they are killing people because they want their organs for organ transplantation. Okay, so you must listen, see what is their concern. Promote the understanding of risk and building resilience to misinformation. You must educate them not to believe in everything that they hear on the net or read on the net, and engaging and empowering communities to take positive solutions. Okay, so that's it, and I hope that it was helpful for you. Any question? Thank you. I think that's actually. بدأ بمن سنويجي أنا إيمان شديد ماله
For the first one, where is it? Thank you. We'll share the Zoom.
Yes, the first one.
No, that's right.
Hello, everybody. I'm Bob. Now this I'm working in many institutions as technology as well. First of all, I would like to thank all colleagues from comstech for the enthusiastic hospitality. And I'm very happy to do this event, of course. As a matter of fact, I would like to talk first about the early event detection. And I hope it will be useful for you. And I would like to help me. What do you think about early event detection?
Yeah, mostly in the early stages and identify.
Identifying the event as early as possible. That helps you actually. Yes, that's it. Let's see the scenario, which is very common scenario for us. For instance,
Oh, this one?
Yes.
Okay,
And you may use the pointer also to change this.
okay. As a matter of the fact, early event detection is a matter of preparedness and is a matter of decision analysis. Let's think about this scenario. For instance, you have encountered an increase of a number of certain disease, for instance, a diarrheal disease. What will you do? I want to come to you, but I have to get stuck here.
identify the cost.
Identify the cost. The other one. Any other idea? This is a very common situation for epidemiologists. Who is epidemiologist here? Please, you tell us.
First, I want to confirm the outbreak and inform the team, like response team.
So you would like to confirm the outbreak. Okay, what about you?
So my screening time doubling
Mhm.
and before capacity building increase definitely and inform to ministries and
Mhm.
take this some and involvement of different teams so like this way.
Okay. I have show you a number of choices. Which one can be correct one? Any idea about this?
Investigation.
Inform the community.
Which one?
Second one, last one. Inform the.
Inform the community. How? Okay.
Number 2, 3, 4.
Oh, yeah.
Second one. All are
Mhm.
The second and last one.
Remember, there is a principle in epidemiology. When you are asked something epidemiologically, first use the phrase "it depends." Whatever you use after "it depends," in 99% it will be correct. So all of these activities can be a correct choice. It depends on the situation. So, as a matter of the fact, it depends on. I would like to talk about in a 2 slides about the principles of decision analysis. As I told you, the matter of early event detection is a matter of decision analysis. In doing, in providing or in deciding, especially in critical situations, time is the most important factor. You usually will be on pressure because of the constraint of the time. And as a matter of the fact that decision analysis is usually based on probability theories. This is an overview of decision analysis. You may be familiar with it. A decision tree, we call it. In fact, in order to perform a decision analysis, we have to do at least 2 choices. For instance, one for the cost of doing some activity and the cost of not doing that activity. This is the important. You have to consider the cost of these 2 different lines. And in order to adopt the decision analysis into action, you have to do all these steps. But in case of a critical situation, such as an outbreak, do you have time to do all of this at the exact time of the outbreak? Usually you don't have. So in that case, early event detection and decision analysis is an ad hoc activity. Before happening of the critical situation, you must be prepared. You must be you must have a protocol for the decision analysis procedure. That is a very important issue. And that's why yesterday was the Global Preparedness Day. Okay. So let's go back to this question again. In case of early event detection for this scenario, what do we need? What is the basic important necessity in order to do an early event detection for this scenario? In order to manage the time.
Inform the community.
Yeah?
Inform the community.
What do you want to do to inform for the community? You don't know anything about it.
Begin outbreak.
Yeah?
Begin outbreak investigation.
Making an outbreak investigation. What is the first step of making an outbreak investigation?
First of all, protect ourselves.
First, protect ourselves. For what? You don't know the causative agent. You don't know even the transmission mode of the outbreak.
Try to investigate the positive agent.
Try to investigate the causative agent, but how? At the first days, at the first hours. I told you, you should tell me the necessity. The first thing you require in order to do outbreak investigation, risk assessment, things like that. Yeah.
Because the case is not known, so try to collect more and more data.
Oh yeah. As a matter of the fact, the information is the most important necessity you need, and you need to have a surveillance system, of course, at the first, as the basis for the getting information, for the collecting the information. And what is the surveillance system? Is that incorrect?
The pronunciation.
Surveillance, okay. Okay, please tell me about your concept, your interpretation of such a phrase for the definition of a surveillance system. Surveillance system.
So it's like systematical approach,
Mhm.
starting with your plans and then collection the data, and then your slide, your stat, whatsoever in qualitative and quantitative.
Okay.
And then your results discuss, and then these results converting to your health policies management.
Thank you.
And then,
What is the cornerstone of this definition? The cornerstone, the important point in this definition.
Systematic ongoing.
Systematic.
Interpretation and innovation.
Again, what else?
implementation of health related.
Promotion of health. This is the important. We do everything. We collect the data, we analyze, we do everything for the promotion of, in order to implement something to promote the public health. If there is no promotion of public health, all the things are wasteless. This is a very important issue because, especially in countries such as Pakistan and Iran, we like to gather information and we do nothing for them. So, what is the drawback of this definition? In case of surveillance of the, excuse me, what is the drawback of this definition in terms of surveillance of a disease?
So they do not tell us the time management, how much they must use the time durations, must because according.
Yes, time is important, the important drawback. But in which aspect?
Collection, planning, management, and implementation.
Oh yeah, you were somehow correct. As a matter of the fact, the surveillance of a disease is based on counting of the patient. We have to be weighted to come to a patient to be presented to a system, for instance, a health system, and then we count it and report it and collect the data. It will take so much time. The drawback is this one, and it hinders their early event detection activity. This is the important issue that the surveillance diseases, ordinary surveillance disease, they in their definition, in their nature, they have. But in fact, we usually confront with a number of situations that we need more rapid response, and we need to detect more the events as rapidly as possible. In that case, we need another sort of system. The matter of the time usually is more important if we are encounter with bioterroristic activity. This may happen so easily. We had such a thing in our country last year. There were intoxication of the school-age girls inside the schools in many episodes. So, as a matter also, for instance, there are some situations that we need to forecast. For instance, in case of the Aedes mosquito and the climate change, and as you know, the global warming helps the spread of the Aedes in different locations, including those locations that didn't have the mosquito, such as in Iran, in the northern part of Iran. Nowadays, we have Aedes, and those locations are prone or vulnerable for the Aedes related diseases, such as chikungunya, such as dengue fever, and things like that. So, as a matter of the fact, early event detection, by definition, is the ability to identify and predict events before they occur, based on the patterns we see in the environment. In fact, we do not wait for a patient to come to our system. We do not wait for the patients to happen, to an illness to happen. And it allows us to take proactive instead of reactive measures to make informed decisions. This is an important issue. So in that case, yeah. Oh, yeah. Let me see. Madiha. Put waiting, remove. Okay, would you please, Maha, let me see your question. Madiha. Oh, yeah. Okay. I can't unmute her. No, I can't do that.
Unmute herself.
No, this is me.
Just try to check.
So as a matter of fact, in case of, for instance, disasters, in case of disease outbreak, and in case of bioterroristic attack, if we have an appropriate early event detection system, we can react, instead of reaction, we can proact. And in that case, we can minimize the cost of that critical situation. As in fact, early event detection is the main tool for preparedness. And the main tool which is used for the for the early event detection. In order to have an appropriate early event detection system, we need biosurveillance instead of ordinary surveillance. And you can read the definition of biosurveillance. Read it, please. And give me your perception. Give me your idea about this definition. What do you think is the difference, the main difference of the biosurveillance and surveillance itself? It's very difficult for me standing just here. And usually my classes walk around and ask everybody. Do you have any idea about the difference between biosurveillance and surveillance?
Does include all the animals.
Yes, as a matter of the fact, the definition is more inclusive than the surveillance. Instead of just collecting data on the event itself, we try to find information about the risk factors, about the hazards, about everything which may be related to the event. In that case, we can say that in that case, we can predict what may happen in the future. In fact, the purpose of biosurveillance can be an early event detection. It can monitor the trends of the what? What do you think? About the risks. We can follow the trends of the risk. And of course, the early event detection and monitoring the trends can give us the situational awareness. In this case, if an outbreak is going to happen, and by the ordinary surveillance system, we can detect the outbreak in here. If we use appropriate biosurveillance system, we can, for instance, detect the outbreak in here, because we are detecting the environmental changes. Let me give you an example. Yeah.
There is a condition that is caused by a rare infection or a known pathogen, and we don't know anything about that. So how we will detect that by biosurveillance?
I will give the example. Okay. Just wait. Thank you for your nice question. Let me give you an example. This is an example suggested by Chinese scientists for the next pandemic preparedness, of course, of the respiratory kind. They try to collect the information of the people who talk with the cell phones, the voice information of the people who talk ordinarily daily with the cell phones. And they train the computers by using an artificial intelligence. They train the computer, and in case of hoarseness, they detect the probable cases of acute respiratory infection. And in case of increasing of the suspected patients of ARI, acute respiratory infection, they can say that something is happening. This is a very, very intelligent sort of biosurveillance system, as you see. They don't wait for the patients to come to the health centers. They don't wait to the patients to come to the health centers and being reported. They just, by using just telephone, their own cell phone. Apart from the privacy or things, ethical things like that, you see that this is a very important example and can be easily implemented in many countries, I believe in your country as well, in Pakistan. Can you give me a practical example of biosurveillance in your country or in your setting you're already working?
So AMR, AMR is a recently project, I think so last 5 years. Primary, I think plan within the collaboration of. Yes, yes. So this one, so this is the best example of the biosurveillance.
AMR, human antimicrobial resistance.
Yes, yes.
It will
What is more for the activity?
That project is based on isolated antibiotic resistant gene from different ecosystem. And that is actually related to the surveillance. That is, according to my contact, that is not actually biosurveillance.
But I think so it's involvement of different health promotion and lead detections and even involvement of the animals and human sides. So according to definitions, if you define surveillance and biosurveillance, they are defined. Even the population's divisions health related to human sides and animal. So it's also including the biosurveillance.
As far as I know, for the AMR, I can give the example in my country. In order to see, of course, in case of AMR, for the outbreaks of AMR, I mean, we say emerging AMR resistance, antimicrobial resistance, excuse me, emerging antimicrobial resistance. And in order to detect emerging antimicrobial resistance, we usually follow the prevalence of the ESBL coli. This is the proxy for the AMR as a whole. On the other hand, we can follow genetically the pathogens. And in case of certain sequences, we can say that AMRs maybe happen. That's right. Would you please give me some easier examples, which can be used daily in the settings, in the health settings? For the biosurveillance. The trend of selling OTCs, over-the-counter drugs. For instance, in the pharmacies, we can use the trend of the selling acetaminophen or adult cold, or all the antihistamines usually used by the people for the common cold. This can be assigned, the increase of the selling can be assigned for the ARI, for influenza, common cold, and things like that. The absence of the school children can be assigned for the future, some sort of disease. The contents, the analysis of the social media contents, for instance, searching people of what, which may be important regarding some sort of diseases. Meteorological data, for instance, for the global warming, we expect the expansion of Aedes will increase for instance, in our country. And according to the increase of the Aedes mosquito, we expect the number of dengue fever, for instance, in certain parts of Iran will be increased. So in that case, we usually, in order to have a biosurveillance system, we usually collect the data from human, animal, and agricultural sources. What this slide remember you?
One day.
Yeah, exactly. As a matter of the fact, biosurveillance is an aspect of One Health approach. Or specifically for the biosurveillance, this is an aspect of all hazard approach. In this slide, you can see the trend of the searching the cough by the Iranian. I have got it from the Google Trend. This is just before official announcement of the COVID-19 in our country in 2019. Here is, for instance, the summer of the 2019. Here is the fall, about November. And here is the January, February 2020, when they officially announced the occurrence of the COVID-19. As you see, there was an increase of the survey in Palestinians cough. Maybe this is similar to Pakistani, they say, survey. In fact, as you see from here, which was about the early months of the autumn, of the fall, there was an increase in the number of searches of the cough among people in Iran. Of course, this notch is related to a difficulty in the internet in the whole country. This is an artifact. And in another slide, I would like to show you that, in fact, the places mostly the people were searching the cough was Kokibuye, Home, and Markaz Promise. And as Kehan said, Home was the epicenter of the COVID-19 in our country. So this was a very important issue. Of course, this is an example. I would like to say that we can use the Google Trend for the biosurveillance system. This is very, very primitive, of course, sort of action. I told you the main difference of the biosurveillance with ordinary surveillance is instead of using the outcome data, we use the risk factor data for biosurveillance. And another issue which is related to the analysis of the biosurveillance data is the fact that we usually use different statistical methods. This is very important one. In fact, we usually use statistical process control methods. Do you know the SPCs? Statistical process control methods are those methods usually used in the manufacturers in order to find the defect product. A modified method is used for the biosurveillance in order to find the events which may happen in the future. The method is somehow, it's a bit complex, more complex than ordinary ones.
Oh, yeah. Well, this kind of methods are used, for example, to monitor the temperature of the freezer, the lab or the industry. And so you have some normal fluctuations, but then you have some peaks, and maybe, for example, in 3 days, you see that you have passed the acceptable threshold. So this shows that something is wrong in the process. It is the same with this method that Trish says. So you have an increase in, for example, diarrhea patient, and then decrease, and then some fluctuation, and suddenly you have an increase. So these techniques show that this is significant. Sorry.
In this slide, I would like to tell the principles. The first principle of the biosurveillance system is that design should follow or should be according to the purpose. In fact, let's go back to the outbreak investigation. In order to use an epicurve, you have a line which shows the endemicity. This goes up, and that shows you an outbreak has happened, and goes down, and the outbreak will be ended. This is a very simple one. In Farsi, we say that, of course, in theoretically, it's very simple. But in practice, we see the epicurves like this. In that case, what do you think that if you are here, is that an outbreak? How do you say that? How about here? Is that an outbreak? It's spines. I told you, first say it depends.
I think it is, I think in this, program, the 2 points. Lower and
Upper.
high.
Okay, you have both of them. Which one do you consider? You have, in just 2 days, you have an up and a higher, and you have a lower.
And this was medium, but,
You take the average.
like line is the medium between the.
That can be a clue, yes. This is as a matter of the fact that this is the difference between the ordinary statistical method and SPC, as I told you in the previous slide. In order to interpret this epi curve, which is completely ordinary one, you have to do some smoothing, and after that you will have to interpret the curve. In fact, the interpretation of the curve depends on the threshold you may design. For if I design the here as a threshold with F limit, all these show an outbreak. If I put the threshold here, this one, for instance, this one and this one shows the outbreak. Putting the threshold depends on the objective. If I want to detect a bioterroristic attack, usually I have to consider lower threshold. If I would like to detect the outbreaks, I have to consider the upper thresholds. Finding the appropriate threshold has its own methodology. Not just counting the average and things like that. It has its own methodology, is somehow a bit complicated. Okay. Another principle for the early event detection is the KISS principle, we say. KISS means keep it scientific but simple. We have to do, or we have to implement the simplest statistical methods in order to analyze the data related to the coming out from the biosurveillance system. And of course, in order to implement an appropriate biosurveillance system, we usually encounter a number of challenges, including data quality. Because we have to collect the data outside the health system. And the quality of data in that case may be different from our own data, which is already coming from the health system. There are many false positive cases. My friend asked me about the uncertainty about the diagnosis. You are right, because in the case of the biosurveillance, we usually depend on the probable cases. And in that case, there are many false positive cases. And of course, as I told you, the complexity of the analysis, the complexity of the collecting data and analysis is somehow higher in the case of the biosurveillance system. In order to have a biosurveillance system, we need an integrated data collection system. We need appropriate legislation system. And of course, all the things should happen in the electronic platform. Thank you very much. This is my first presentation. I have used these two references in order to prepare my slides, and for more information, you can read these two books. The slides are on the desktop of this computer in the folder of day 3. Any question? I'm at your service.
Thank you very much for this. I think now is the time for break.
Oh, time for break?
Yeah.
Okay.
We'll come back for 1 hour.
Okay, thanks.
Break is not 1 hour. Yeah. 15 minutes is enough.
So the break is about 15 minutes.
این مصاحبه رو چند نمت میگیریم.
به نام خدا خانم رایگان هستم، مصاحبه اپیدمیولوژی در این دوره برای بررسی و مدیریت بحران در خدمت دانشجویان پروفسوری بودیم که داشتن از دانشجویان به صورت آنلاین به صورت حضوری شرکت کرده بودن و میتونید از فکر کنم شما میتونید مبحثی که من ارائه کردم انواع مطالعات اپیدمیولوژی و کاربرد اینها در مدیریت بحران. می‌تونم که
در خدمت من برای پاسدار هستید برای برگزاری فعالیت بحثی که من در خدمت اختصاص می‌کنم شناسایی زودرس ناشکران و مباحث مختلف سلامتی بود و بحث‌های اخلاقی برنامه‌نویسی مدیریت که اینجا برای تشریف دارم از کشورهای مختلف پاکستان، افغانستان و مجازی منتظر دو دوره رو در واقع ایران برگزار کرده
Thank you. Okay, thank you very much for your kind participation, for your kind and active participation. Thank you very much. The next topic I would like to talk about is outbreak plan. In fact, outbreak plan is the matter of every organization which is involved in outbreak management. And of course, the objective of the outbreak plan is to define the roles, the resources, and the responsibilities for the outbreak management for each organization separately, of course. And as you know, many organizations may be involved for the outbreak of for the management of the outbreak. Let's say, what do you think an outbreak plan is? Is that the same as outbreak investigation procedure, or is that a different one?
I think, regarding different situation.
Different. So what it is?
It is according. Different situation.
It is about of investigation. The plan, the part of the outbreak investigation.
The part of outbreak investigation. Okay, so if the outbreak investigation is actually bigger, and the plan is inside the outbreak investigation. Okay, what's your idea, sir?
This plan is made according to the data and the information that we collect from the outbreak investigation.
Oh, yeah. So in that case, this is a part, a component of outbreak investigation. You mean?
Next to investigation.
Next to investigation. Any other idea?
Detection, investigation, response, and evaluation of information.
So which part is the outbreak plan? All of these are components. Okay. And
Plan of the outbreak, control and treatment measures.
thank you. It's the most nearest one. In fact, outbreak plan is the context for the outbreak investigation. If you don't have an outbreak plan, you cannot have an outbreak investigation. See? So in fact, as I told you, outbreak plan encompasses the entire outbreak management process. Part of it is outbreak investigation. You know, in order to manage an outbreak, we do many things, including outbreak investigation. We do risk assessment, we do control measures, we do, for instance, communications and things related to risk communications and RCCE. And all these happens in the context of an outbreak plan. If you don't have an outbreak plan, the response and the other parts will not go appropriately. So, again, outbreak plan shall be prepared before the outbreak, the occurrence of the outbreak. It's a matter of preparedness again. And this shows again the importance of the preparedness. And if we see in this slide, in fact, this is the management framework of an outbreak. It has many different components, including preparedness, including surveillance, including the outbreak investigation. As you see, outbreak plan actually are the activities related to each component of this framework. So for each part, I would like to give the explanation that a plan means. For instance, let's go to the preparedness. In the phase of preparation of an outbreak framework, the plans are related to the development of the outbreak protocol. In that case, we need to identify the outbreak management team. We shall have a distinguished person in order to have the outbreak management team. And they should be able to cover all the scenarios related to outbreaks. For instance, you in here can be the outbreak management team. The other part of the other component of the outbreak plan related to the preparation phase is designation of outbreak coordinator. Who is the coordinator of the outbreak? Usually in our country, we have a team, an outbreak team in our country is consisted of two main persons. One is the disease expertise, and the other is environmental health expertise. And of course, with the help of the laboratory, they make a team and they go to the field and do outbreak investigation. They have to be trained and they have to be updated periodically. Another issue related to the preparation phase is assembling material necessary for outbreak investigation and response. In order to send the team to the field, we need a number of materials, including different logistics, for instance, automobile, for instance, I don't know, laptop, things like that. These should be considered in the outbreak plan. The preparation of them shall be considered. Identification and addressing the training needs. This is another issue related to the preparedness phase of the outbreak management framework. And the other part is the surveillance system, which is related to the diagnosis of the outbreak. In general, we have two ways to detect an outbreak, two tools to detect an outbreak. One is indicator-based, which comes from the surveillance data, routine surveillance data, or biosurveillance data. Of course, one type of biosurveillance data is syndromic surveillance data. We usually in Iran nowadays, especially after the COVID-19, we use syndromic surveillance a lot. And another method is event-based detection. What do you think about event-based detection? What it is? Event-based method for detecting an outbreak. Do you know that?
That's practical events based.
Yes, yes.
Depend on the practical in the field.
Mm-hmm.
So any suspect anything happened
Oh, yeah.
in healthcare or hospital, this should be inform the responsible person.
Yes. In fact, for the indicator base, you are dependent on the numbers, on the number of patients. But for some diseases, for some events, you don't have any surveillance system to collect the data and produce the numbers. In that case, you are dependent on the events. In Iran, what we have is a system, a phone call system, a network of people. In case of suspecting any sort of outbreak, they call to the central system and they tell that something is happening. Confirmation of an outbreak is another thing. The response team go to the place, to go to the field, and in case of improvement, the outbreak investigation and things like that continue to happen. So we have two kinds of detecting systems in order to detect the outbreak. One is indicator-based, the other is the event-based. Remember that event-based detecting outbreak is usually a very late one because we have to be waited that event happens and be detected and things like that. Another issue related to the confirmation phase of an outbreak is in fact, for the outbreak plan, is the regular examination of the surveillance system to be sure that the increases of the disease can be detected as an outbreak. This is an important issue. We have many lessons learned after the COVID-19 in order to implement the surveillance system. We used to collect the data, but the analysis was not appropriate. And usually we missed a number of events. We have to be ensured that the analysis of the collected data is appropriate, and this needs some sort of protocols in order to be implemented. As I told you, use the statistical methods, use appropriate softwares and things like that, all goes to this component. Another issue related to the confirmation of an outbreak is maintenance of good systems to receive and evaluate the reports of the outbreaks from local centers, including professionals working in the local areas. In the phase of the scripting of the outbreak, we need to be assured that the team who is responsible for the outbreak management and for the outbreak investigation is able to do outbreak investigation appropriately. In fact, in order, the team should be able to collect the data appropriately to develop the case definition. The case definition is the most important part, because this is actually the this is the tool that provides the ability to count the patients, to complete the outbreak investigation, to characterize the outbreak by person, place, and time. In fact, we have to be assured that the people who are involved in outbreak investigation can analyze the data collected during the outbreak investigation process. And they should be able to develop hypotheses. And they should be able to implement the control measures as well. And this, the other part of the outbreak plan, which is related to the investigation phase of the outbreak management framework, is the investigation part. And of course, in that case, you have, before the outbreak, in order to planning for the outbreak management, you have to find the capacities for epidemiological investigation. Usually in our country, in Iran, we do that by the help of the medical universities, especially School of Public Health, especially places such as Pasteur Institute. We use this scientific group in order to do epidemiological investigation. Usually the people who are working as public health staff are not involved in this phase. I believe doing a full-scale outbreak investigation is somehow difficult for ordinary staff of the health. Another issue related to is the environmental investigation. Of course, this happens in our country by doing rapid risk assessment. And all the staff who are involved in outbreak management shall know the methods of rapid risk assessment. And by rapid, I mean that the assessment shall be completed up to 48 hours following detection of an outbreak. Dr. Azad Manesh just mentioned the steps of the risk assessment and rapid risk assessment. And following the rapid risk assessment, we use rapid risk characterization, in which we prioritize according to the matrix, as Dr. Kehan, Dr. Azad Manesh has told us, according to that matrix. And in the phase of control for the outbreak plan, we shall know how to implement control measures. And I believe that Dr. Arash Kia told us about control measures. Yes, you told us. Okay, I usually make mistake between our colleagues.
Yes.
And the people shall know about it. Of course, this is very important part, because the controlling measures shall be based on the best evidence provided by the outbreak investigation. Sometimes we have some troubles regarding this matter, because the health staff goes to the field and they want to implement the control measures just according to the basic knowledge they have, not to the evidences which is happening in the field for the outbreak. And of course, the other one is the reporting of the outbreaks. Of course, this part of the outbreak management usually happens appropriately in our country, but the follow-up of the risk communication, things like that, we have some difficulties regarding this matter. And the last part is the outbreak documentation, which I believe that Dr. Arishkia will tell us more about this part of the outbreak plan. Just for remembering and just for mentioning the matter of preparedness and emphasizing the matter of preparedness, I would like to talk to you about the PRET. Have you ever heard about the PRET, which is an initiative recently proposed by WHO? PRET. Have you ever heard this? Okay, PRET stands for Preparedness and Resilience for Emerging Threats. In fact, it is an initiative proposed by WHO to prepare for the next pandemic, which may happen in the future years. In fact, according to the lesson learned from the COVID-19, which are the importance of preparedness, the importance of health as everyone's health, and of course we have to notice that no one is safe until everyone is safe, and response must be agile and adaptive. The WHO has proposed this initiation. And according to PRET, this is an innovative, which is hazard-based. According to this initiative, the WHO recommend to have biosurveillance system, different biosurveillance system, in order to detect the health-related events as soon as possible. And of course, they do not want to prepare a new one. They want to prepare they want to make the surveillance system based on the existing capacities. They don't like to provide a new surveillance system. An important issue regarding implementation of PRET is grouping pathogens according to the mode of transmission. This is your answer of your question. Answer of your question. At the beginning of the outbreaks, we don't care about the exact agent which is responsible. We care about the mode of transmission. We try to find the mode of transmission. If it is airborne, we usually do a number of common activities regarding management of the airborne outbreaks. If it's foodborne, and other things we'll do regarding the management of foodborne outbreaks. So at the initial phase of the outbreaks, we do not intend to find the exact agent. We just want to find the risk factors and manage the risk factors. As I told you, we would like in the PRET, we would like to strengthen the existing systems and capacities and filling the gaps and incorporated innovations. And the last one is the promoting equity and inclusive coherence. This is the pillars of the PRET initiatives. And of course, I have used these two references for the outbreak plan in order to prepare my slides regarding this title. Thank you very much for your attention. Still the last one. Thank you very much. Thank you. Okay, and the last topic I would like to cover, of course, I hope you are not tired watching me, so
They have just come back
continuously. Yeah,
They have just come back from break.
yeah, oh yeah. It's the matter of ethics in epidemics. It's a very challenging one. let me give you a number of ethical considerations. But before that, I would like to give your idea about the matter of ethics. What sort of ethical problems we may encounter during an outbreak, during critical situations, as a general? Any idea? All the things are ethical? What sort? Yeah.
Religious issues would mostly arise during our arise during our.
Okay, what are they?
Mostly some people, religious scholars says about outbreak that these are around something alarms, but it seems that.
Yes, religious matters can happen during outbreaks. Yes, you're right. Other things. Privacy. Yes. Privacy.
And concern
Mhm.
Like in an outbreak, an infectious outbreak, actually we forget the professional ethics, like wearing mask in a proper way, attending the patients. That's all comes in professional ethics. So we actually we are not dealing with that in outbreak.
Yes. In fact, the most important things that happens during outbreak, because of the constraints regarding resources, regarding time you have to do many things just with short resources in a very short period of time. So you may not be able to consider ethical issues, including religious issues. As a matter of the fact, balancing individual rights and public health problems is the most important issue which happens during outbreaks. Another issue is allocation of the limited resources. To whom shall we allocate the limited resources? This is a very important question, which usually happens.
We should target the beneficiaries first.
Yeah?
Targeted beneficiaries.
Yeah, this may be an answer, You are right, we are going to discuss in more details. And another issue is decision making. Of course, the two previous issues will make the third one, which is the decision making in time of crisis. What sort of decision we would like to take, if you are a decision maker, to impose the least ethical issue. This is very important. Let's discuss about the situation with, of course, ethical issues has many different aspects. And I would like just to talk about the triage. Do you know the triage?
So it is sort.
Yes, in fact, sorting.
Agent, host, and environment.
In fact, sorting the patients
Yeah.
to give them services, to select them for providing services. How do you triage? Consider you are in an outbreak situation following an earthquake, for instance, or in your country, flood. In Pakistan, you usually confront with floods nowadays, and you have an outbreak of diarrhea. Many cases of diarrhea coming to your health center. How do you decide to manage these cases?
I think protect the plan and identify the responsibilities for all the healthcare professionals, and also protect the standard code or color for identify the patient that need more care or more treatment.
Can you challenge this gentleman regarding the answer? I would like to challenge you. If you give service to most severe cases, as you mean, You may change the moderate cases to severe one because you have ignored them. What will happen? Let's change the idea. Service to the most healthier cases. So this can happen so easily. As a matter of fact, whatever you choose, different ethical issues will happen. So, in fact, we have to, in order to manage the ethical issues of the triage, because just I would like to focus on the triage. Many aspects have the outbreak and epidemics, many ethical aspects, but I would like to just focus on because of the constraint of the time, on the triage. A main objective of the medical, this is a fact. We have to consider these facts in order to implement appropriate triage, in order to consider the appropriate ethical issues. In fact, the most important objective of the public health system is to minimize the morbidity and mortality. This is a very general aim, a very general objective. We have limited time and resources. Of course, this is very evident. It's necessary to choose who should be given and what sort of treatment shall be given to him. This is the example I told you. Would you treat the healthier cases or would you treat the more severe cases? And whatever strategy you choose produce its own ethical issues that make you responsible regarding that issues. These are the facts. And of course, in order to manage the resources, we can use triage, we can use rationing. What's the difference between triage and rationing? You know? Triage, as I told you, is somehow sorting the people, the patients, into different groups and by some criteria give the priority to a number of groups. But for the rationing, we don't sort the patients. Actually, we look at our resources. For instance, we have an operating room, can provide services for 10, 12 people a day. So actually, according to this capacity, we provide services. We don't use any sort of priority for the patients. If the limit is reached, we can stop providing the service. Sometimes we use both of these management strategies. And according to the topic I would like to type, the triage has 3 levels. The primary one, which is implemented at the center of the emergency. The secondary one is implemented in the emergency room of the hospital. And the third and the tertiary level is implemented, for instance, in a ward or in the operating room. Each level of the triage, the ethical issues of each level of triage may be changed according to the nature of the decisions we make at each level. And of course, in order to ethically justify our activities regarding the triage, we made the criteria of the triage may be different, and the criteria may provide different ethical issues as well. For instance, if the aim is saving the greatest number of people, we usually give the service according to the prognosis of the patient, as you just mentioned. If we consider the time of coming to the health services, we provide the service to the first people who come to our center. In that case, the first come is the first one who will be served, and give the priority allocation to patients who have access to our service. In this case, the assumption is that all people have the equal accessibility to our service, to our provision of the healthcare services. Another criteria for the triage is protecting the most vulnerable people. As you just mentioned, that we may provide a number of services, for instance, for old people. For the first one, if we implement the greatest number of people, in that case, we may lose a number of vulnerable people. For instance, old patients may be neglected. But in this one, we may lose a number of healthier people. If we are going to protect the most vulnerable patients. Another issue is, or another criteria for the triage is using equal access method. In this case, we would like to provide equal access to many people, to as many people as possible, and not just who have access to the our healthcare system and things like that. Usually, for this strategy, for this criteria, we provide healthcare services randomly to the people. Actually, in some settings, we can use this sort of criteria, which has least ethical issues as well. And we may, of course, the worst one is this criteria, priority for the most important people. In fact, in this case, we may provide healthcare services for those who are mostly affect the community, for authorities and things like that. This is the least ethical criteria for implementation for the triage of the patients. And of course, this is mostly based on individual judgment. This is the worst one. In order to consider the ethical issues regarding for the triage criteria, as I just mentioned, we have two different categories principle in order to implement the ethical issues. One is substantive, which is related to the nature of the criteria, to the strategies we would like to implement. One is consideration, but in fact, these principles, this sort of the category of principles, considered to explain why a particular policy or course of action is ethically justified. And the other is procedural. In fact, these are related to the way of the implementation of the ethical issues. In case of substantive ethical values, we have to consider individual liberty. This is very clear. Protection of public from harm, and proportionality. Proportionality means that the jeopardizing the ethical issues should be minimized as possible, as much as possible. And privacy, which you have just mentioned, that in an emergency, it may be necessary to, because of the public health issues, it may be necessary to jeopardize their privacy, but it shall be go back and the privacy issues shall be regarded as soon as possible. Another issue is, which is related to substantive ethical values, is duty to provide care. In fact, healthcare providers will have to weigh the demands of their professional role against other competing obligations to their own health and families and friends. This may happen so frequently during critical situations, because the healthcare providers have their own families, they have to serve for them in order to save them from the critical situation, and they are obligated to providing healthcare services. There is a trade-off for doing this against doing that. And reciprocity means that society support those who face a disproportionate burden in protecting the public health and to minimize the burden as much as possible. this usually happens for us in our country. In order to protect our healthcare system, we usually, in case of the critical situation, we usually provide a number of services for our healthcare services to be assured that they can provide they can provide their activities during the critical situation. And another issue, which is equity, and you know that during a pandemic, you have to make difficult decisions, and difficult decisions usually may ruin the matter of equity. Another issue related to substantive ethical value is trust. And as a matter of the fact, trust usually increase or promote by appropriate risk communication, appropriate communication, in fact. If you inform the society, the community appropriately, the trust will increase, and the people will, in case of appropriate trust, they can be empowered as soon as possible, and they can help you for the management of the critical situation. The other one is solidarity within and among healthcare institutions to help the people to empower to manage the critical situation. And stewardship is in fact, ethical behavior and good decision making produce an appropriate stewardship for the people. The procedural values are reasonability of the decision made for the people. The other is the open and transparent decisions, and the decisions should be inclusive as much as possible. And of course, the people who are making decisions should be responsive and accountable. And of course, accountability is not just being responsible, but being trustful in order that the people can trust you. In conclusion, ethical issues are very, very challenging according to the situation, according to the type of the crisis, according to the type of outbreak we are confronting with, and it needs consideration. And in fact, we have to promote it because of different situations will require different ethical issues. Thank you very much. I viewed this booklet from WHO, which is Ethics in Epidemics, and just one aspect of the ethical issues in the epidemics I have just noted. So, thank you very much.
No, it's the flash. Okay, assalamu alaikum again. Ashhadu an la ilaha illallah. Okay. Well, probably we will not have time to for Dr. Ashkia to talk about the documentation. So maybe this will be the last session of this course. Okay. So two subjects that remained for me. One is agency roles for outbreak management, and the other is the privacy of data. I do not want to talk about them too much because for different reasons. For the first subject, this is very dependent on the system in your country or your region. So if you are in the United States, it's different from here, and it's different from Iran. But again, going back to this slide, you see coordination is the key cornerstone of this chart. So it is in the middle of everything. And you have to communicate between other agencies. You in the health sector most probably are not responsible for or in charge of, for example, putting in place the quarantines. You cannot control the movement of people in the country. So you have to talk about this with other agencies. So for this, first of all, you should list the agencies who are responsible in your country for different activities in this chart. So I don't know how it is in Pakistan. For example, in the United States, if there is an outbreak and it is limited to a county, okay, maximum the different organizations in the same county are responsible, so they have to be coordinated. If it is more than one county, then two municipality probably should have to be in touch. If it is higher, it is the state level. If it is higher, it's the federal level. In Iran, the system is vertical, so almost any problem goes first, or not first, but the capital is aware of what's going on in a city and around the country. Though in our system, all the country has been divided to 64 universities, medical universities, which are responsible for both health of people in their area and also medical and related education in that area and medical related education. So sometimes they can handle it at the university level, but anyway, the capital is aware of what's going on. Sometimes they just have to coordinate with the governor, and sometimes it's much higher. So you should have your own list of stakeholders. And you see, these stakeholders differ depending on the situation. For example, if you want to control the ports for entry of, I don't know, foreigners, for example, or the ships with some goods that you want to disinfect them first. So you have to ask for the partnership of I don't know who is in charge of organizing the ports in Pakistan. Or when you have a lot of goods you need to transfer them to other cities to deliver them to the health system, you need a transportation system. Or even more prominent than that, if you want to place a quarantine on a city, so you must be able to provide them with food, with oil, with anything that they need. And if you lock down the city, first you need the military, who is the army or who is in charge of that to help you. And second, you need, for example, the transport system to provide you the trucks or whatever is needed to deliver the goods inside the city. So it is a really complicated issue. And considering that nowadays air transport is very common, so airports is also an important point of entry in outbreak control, especially at the national level. So just find out who is responsible for what part of that chart. Then there are some issues within them. These agencies usually do not work well together. So you need a lot of motivation to make them work properly. And the most important than anything maybe is the trust between them. Each of these agencies are usually willing to be named and to use this situation as a propaganda for what they are doing. So we are very important, and our role is critical, things like this. So you must understand them. And well, we are not here to bargain or to discuss about who is more important. We are here to help people. So whatever it takes, usually you should do that. Give them the credit and gain their trust. And to tell you something, sometimes also you need some bargain chip. So consider that too. Just one occasion, we were running out of some extraction kits in our lab during COVID-19 early weeks. And I knew that there are a lot of these kits in a warehouse of the Ministry of Health. So we asked them to give us something like 20,000 reagents. And they said, okay, but I didn't receive them. So I asked them what is happening. And they said, who is going to pay for that? Then I got angry. Hey, man, this is the Ministry of Health. We are working for the Ministry of Health and we are working for the people. And we have this kit in the country. So who should pay? I don't care. So just give it to me. Anyway, they sent the kit to Tehran and put it in a warehouse, but didn't give it to me. So luckily that same day, we received a batch of samples from the cabinet, from the government. So I told my staff, don't release the results until I tell you. Then I called the director and said that, okay, I will not give you the results unless I receive the kits. And just maybe in 1 hour, he managed to do the arrangements. And by midnight, Arash and I took my own car and went to the warehouse and packed it with the kits and came back. And of course, we gave them the results. So you must see also your bargaining chips. Sometimes this is needed. In addition to that, there are some international organizations which are responsible at some levels. Well, for some diseases, they should be notified at the first place. These are the diseases which are reported for IHR. So usually your IHR contact point, the national contact point should be aware of that. He may or she may contact WHO, and they may give you some advice or some help. These are also some obligation. I mean, this is not just a matter of preference. Some of them are obligatory, so you have to report them. And also, they provide services. During early days of COVID-19, we received a group of experts from WHO to help us, to review us. Well, by helping, it is not a bad thing. Or sometimes you are in the middle of a fight and you do not see what the others see. So it is good for you to have an observer from outside to see what is lacking in your system. So they came and fortunately, I was in charge of the laboratory and they were very happy with the laboratory system. And now IHR is under review in the WHO. So probably the next version would be a little bit different, maybe some post rights and duties of the countries may differ from now. So you must be aware of that in the near future. There is another international attempt to prepare a pandemic accord. This is some kind of convention, a legally binding convention for countries based on our experience in COVID-19. Well, countries have different position on that. I don't know what's the position of Pakistan. Right now, we do not prefer it. We prefer to enhance and improve IHR. But anyway, if this happens, this is, for example, supported mainly by African countries because they are seeking support of others. And considering what Babak said about the mottos of preparedness, the initiative, nobody is safe until everybody is safe. So they want to make sure they receive the support from rich countries timely to be able to respond well and then protect also the others. So we must see what's going on and what will happen in the near future. And something very important, you need a lot of advocacy. This is the skill that maybe you lack, you as healthcare people. So again, going back to the structure of the team, this is a teamwork. You need to have people who are able to advocate for you, to convince the others, and do the arrangements with them. And even at the diplomatic level, So sometimes you need, again, to give them something to receive some help that you need. And these are, well, you can learn them by yourself if you are, I mean, if you are that kind of person that is good at that, or more preferably, you must have someone with these qualities in your team. Well, there is a definition, I mean, the concept of the public health emergency of international concern in WHO, which is the key point of initiating an international response for that. And we have had a few of these fakes in the past 10 years. Zika was one of them. I think polio was one of them. COVID-19 and SARS-CoV-2. SARS-CoV-2 is COVID-19. The other one, MERS, I think was one of them. So these are of international concerns. So you need to arrange with WHO and with other neighboring countries. Maybe you have to exchange your samples with them, your experience with them. You must have the permissions from, I don't know who is responsible for permitting the export of biological material in your country, or receiving the helps from the others, etc. But these characteristics are also helpful for you in a national level. So a fake is an extraordinary event which is determined to constitute a public health risk to other states through the international spread of disease and to potentially require a coordinated international response. These are the specification of such an event. It should be serious, it should be sudden, it should be unusual or unexpected. So if you have, maybe polio, I don't know, do you have polio still in Pakistan? We have in Afghanistan. So probably, well, if it is contained in Pakistan and Afghanistan, maybe it's not a fake. But if it goes to other countries during, for example, Ziyarat Arba'in to Iran, to Iraq, that's a fake effect. It will trigger the criteria of fake. So these characteristics are important for finding an outbreak in the first place. And also, another aspect of this organization, this WHO especially, is that it provides some tools for evaluating your preparedness. And some of them are done by yourself. You do not need to, and you don't need a foreign observer. Some of them need foreign observers. For example, this is one, SPAR, which is a self-assessment annual report. So the country evaluates, I think, 35 core capacities for these indicators, sorry. And 415 RHR core capacities. And then they send a report to WHO. This is an indicator of how prepared the country is. Or better than that is a joint external evaluation, JEE, which is done by a group of experts who come from WHO. Of course, your national experts gather, you collect the information that which are necessary. I think now it is 19 areas, I think. So you collect the data, they review it, and then they give you a score. And if you look at the list of these 19 areas, you will see very interesting points. For example, legal instruments. Does your country have enough legislation to prevent an outbreak and to control an outbreak? And what is the control measures in your country? And well, probably it also includes the legal issues related to ethical aspects that just Dr. Esheti mentioned. Do you remember that during COVID-19, some countries, they decided to separate the old people from the respirators, because they had not enough respirators and they preferred to give it to young people. So this is a matter of ethical issue. It is a big ethical issue, in fact. So you must be ready to decide in those situations. Again, for financing, for example, for antimicrobial resistance, for zoonotic diseases, food safety, biosafety and biosecurity, immunization, then to detect, these are capacities to detect the disease. What is your national laboratory system? What is your surveillance system? and what are your human resources for that. Then in the response phase, these are health emergency management. Do you have it or not? Or what is this? This is status. Linking public health and security authorities, health service provision, infection prevention and control, risk communication and community engagement. And also in this is also very interesting. For example, what is the coordination between point of entries and border health? So you see that even in this evaluation, WHO is looking for the connection of your health system with the legal system, with the, I don't know what is the responsible for point of entries, police or other authorities. Secretary of Health Establishment. Okay, and who is I mean, they should have a legal arm too. So who is who? I mean, a law enforcement tool should be there also. Who is doing that?
Airport and security.
Is it a part of the health system or is it part of security?
Well,
okay, that's interesting. Okay, and even what do you do in the case of chemical events and radiation emergencies? Especially for chemical agent events, it is important that many of them may mimic a common source outbreak. So the first step would be very similar, just the detection. When you detect this as a chemical outbreak, so the course of measures that you take is very different from the infectious disease. So you see, they are looking for this coordination also inside your country. And this is a good guideline for you to follow to see what you should do. Well, the other part that I want us to mention briefly is the privacy of information and data disclosure policies. This is basically a matter of ethics again. So like what Dr. Shati mentioned, you must have a clear view of what you are doing. You must make a wise decision and based on ethical principles. Usually these are done by a group, by a committee, so it's not a personal decision. And it is the balance between the public interest and the personal rights. So you must justify it. You collect information. For example, when you are doing an outbreak investigation, you collect the name of people, the location, their family member, things like this, and these are personal data. Or in one case, for example, the national unique national identifier or something like that, if you have it in Pakistan. I assume you have, yes, I know that you have. And you do this, you must maintain the trust between people and you. So they must not feel that you are violating their own rights. Well, these are some principles for that, should consider them. Personal data and confidentiality. Well, in some cases, it is possible to make the data confidential. So you do not really need the name of people. You just do a blind, for example, sampling to see what is the prevalence of one disease or one agent in the community. It is not necessary to collect the name, so don't do that. But never go for extra data. I mean, this is not good. And it's important that what is your legal framework? Does it allow you to collect those data or not? And if it is not, people may take you to the court, and they're right anyway. And when you collect the data, you must be transparent and ask for the consent, inform them, and then get their approval to collect the data. And try to minimize the data that you collect. So as less as possible that you can. Don't go for extra data. You know, extra data is a huge burden. And you know this, when you may get lost in the data. So it is not an advantage for you. And in some cases, you may collect the data with their name, but when you send them to lab or to other agencies, you may omit the names or give them pseudonyms or give them codes, etc. Yeah. And one very important issue is now the security of data, because mostly we collect the data on computer servers, so they are vulnerable. Hackers may attack them, and then they steal the data. They may blackmail the government or whoever is responsible for the data. You must have some what is it? I do not remember the word. You want to have force in it. Anyway, what are we, a risk assessment, risk management plan for that. So maybe you need a backup of it somewhere else. You need its security to be enhanced and things like this. And then the data sharing, again, other agencies may look for the data that you have, but people trust you as the health sector, and they do not have the same trust in other sectors. So you must make sure that you're keeping the rights of people and keep it in their privacy. You do not give them their data very easily to the others. Maybe they do not care about it. And have an incident plan. So what happens if your data is lost, if you are under a cyber attack, things like this. And the situation is now more complex because of these new challenges. During COVID-19, many countries developed softwares to monitor their people, their movements, their health status, their testing status, etc. This is an example in Iran. We had this mask application, and it was very helpful, but it collects your personal data and even more than previously, because it collects some metadata, your location at any point of time, for example. So the amount of data that now is possible to be gathered is much more than before. And these are new challenges. So what are we going to do? These are the topics which are now addressed by researchers and by public health officers. This is one example. So what a smartphone application, which policies did they take during COVID-19? And I think that was all. Let me see. Oh, yes. Okay. So that's it. We're coming to the end of our course. I hope you have enjoyed it, and I'll give it to Sana to give you your closing remarks. Or do you have any questions for me now? Don't you want to, okay, you want to give them posters, then talk about what they feel about and what they know. Okay. So
running sample, for example, COVID, in APU, we have collected the sample to assess the COVID prevalence in pregnant female and then vaccination whatsoever. So I have looked at ethically, if we want to find another assessment like proteins and variants, so it's allowed to use the same sample for assessment of the same, remaining already stored.
Most of them are not going to be in this dilemma as your ethical committee at the university, because this is very important. You cannot do beyond what is approved by the ethical committees of your institution. and they are responsible for the trial.
Yeah.
So in some cases, maybe they approve it by some measures, for example, anonymizing the test, for example. Maybe they have some, they offer some trade-off between something and something. Something, for example, you have less data, but you have, for example, if you for example, need to follow, if you don't need to use the sample, maybe the easiest way is to anonymize them and then use them.
Yeah.
Okay. Usually, in these cases, you should have a prior consent of the patient who have collected the sample. So you have four, in which you mentioned that this sample might be used for further investigation in the future. They are this is the idea. But even if you don't have it, that's the job of the ethical committee.
Yeah, exactly. Actually, I am also member of the AQ ethical committee. So I have like one or two from my colleagues. They are assuming like quality level or I don't know physiology. So they are considering me like what is the further investigation step to use the same sample. So because I don't know another PI. So the main I have already, if you have already consent form, you have mentioned this line, we are further and then we will wait. Otherwise, you have not allowed even you have take again the concern about the ethical committee. So if you have done the first this step within the consent form, then it will be allowed and otherwise it will be disqualified.
I'm not an expert in it, but issue.
Yeah, I am not expert, but it.
What there are some criteria. What we tell you, for example, in one occasion, this is not necessarily relevant to our talk, but it's interesting. I know someone who wanted to do a clinical trial in the UK for a new treatment for a cancer disease. And then he asked for the end stage patient to be this treatment be applied to them. And this is interesting. The ethical committee decided that you should not go for the end stage. You should go for the stage before that. Because the end stage patient will not benefit from this treatment. But the previous one may benefit. So they in fact, gave the PR more flexibility on choosing the patient. So they facilitate what he wanted to do. So this is what they decide. I don't know. But anyway, sometimes it is a matter of, it's a matter of emergency, it is a matter of how to get the health insurance. So they accept to do that in those cases.
That's mean we don't have any standard criteria regarding ethics. Like it varies from country to country and station to station and so on.
Well, I think so. I think I'm a medical doctor, but this field I think is somehow new. So we recently have some PhDs in medical ethics who give us lectures on what we should do. And I'm not sure even if we have any professor, any full professor of medical ethics in Iran or not. So they are very junior. They started very fresh.
But if you have conduct international clinical trial within the collaboration of another country, you so Islamabad IBR, International Board, I think so. IBR, sorry, I don't know full abbreviation, but already have done department in Islamabad International when you have a collaboration with another country. So, for example, in AKU, when you collect the from AKU, and if you have an hospital, then you have, and then you have also have to prove the IBR, and then even from another collaborator from your University of Columbia. I have recently project this is collaboration with University of, so you have to even get the assessment. Sorry.
You are saying that first you need a approval from IBR.
So first your institutions, then for your collaboration and even your hospital, and then IBR. So IBR is the final stage, Pakistan, when you have a collaboration with an international universities. So we have a
So you still have some time for doing the post-test. So we would like to know your opinion about the course, how it was. And we appreciate if you're frank enough to tell us really what you think, what you wanted, what you expected, what you received and what you expected and was not met during this course.
How we can improve it for the next level.
Okay. Yes.
First of all, we introduce ourselves. We are IMM students. So we are happy and opportunity to participate in this course and have a chance. Thank you for commenting and thank you for your institute to give us the opportunity. Thank you very much. We have many experience and information as optometrists. And inshallah, we have a benefits when we come back to our country. Thank you very much.
Okay, any other comment? The critics, especially.
It was a very great session. It's a very great session, and one of my colleagues who have been told me about this book. The title is very interesting. And I want to highlight one more thing, I'm a public health fellow in Health Services Academy, and there are so many colleagues here. And during our all lectures, all our core faculties always mention your country, Iran. Like Iran has a very strong public health system, very strong preventive health care. Like even the other developed countries as well. And when Pawan told me this course was conducted by the Iranian colleagues and Iranian team, I was so enthusiastic about this course, and it was a great session. Thank you so much.
Okay. Yes, lady.
Session, no doubt. We have learned a lot of different vocabularies, like IHR, WHO, Similarly, you told us about the pandemic record, and definitely I would like to read in detail about this pandemic record in the countries what the part of the record. I have learned a lot of things related to epidemiology because I had been reading community pharmacy once, but I had to do a lot of effort to capture some ideas related to epidemiology, but maybe comparatively easier for us. One deficiency that I felt was the handout to the patients. You should hand out for all my patients. We take the notes, then related slides.
Okay.
So I think the next sentence, whenever you can message some kind of standard, handout for the patients. Because that was a term pre-test and the post-test. But we don't have anything to prepare for both pre-test and the post-test.
I just want to if you want to sort of paper form, even you can a digital form and soft copy of.
Paper form is better. That's a very good suggestion. Because we can write down your own uptake of just like in the side.
And also, usually here, some investigation, like I found that it can. So I was given writing well, but I did not know what is that in the home. And the addition.
The main purpose of you want to. So for this printed, the main purpose of for the printers was to have for us to have an evaluation of what level you are, and for yourself to have you see you have the same questions. So to see how is your answer different from the beginning or not, and it's also important for us. So it's not something very serious. It's not an exam. And we will give you. Pardon? We will give you the additional form of the slide right now. But hands-on before the session is very correct.
I know different classes, 70 classes. So it's great session. Thank you so much. And I have already observed your health policies and your investigation program. So it's great time. Great time to learn these activities and yeah, it's learn more responsible. Your precautions, your steps that you have already used, and it was great. Thanks so much.
Okay. Anybody else?
Yes.
Yes.
Being a student of undergraduation level of public health, it was a great opportunity of learning for me. And besides that, also an exposure and networking opportunity with all the colleagues for I had good interaction with friends. And overall, I will just say, overall, it was very good.
Thank you very much.
Also, I have just one suggestion. So your collaboration is like one hand. So in Pakistan, this one hand is not prominent. So it's a just suggestion. You can also upcoming within the one hand collaborations like these ones training program. So it's will be great benefit for all, yes, for all the Pakistani students, because there are just you can say that just learning few, even if they are later training courses that regarding the one hand, and your approach is fully one hand. So I request you upcoming within collaboration of the one hand training session. So it will be great for Pakistani students, because it's upcoming one hand and they concept. So it's amazing, and you should also, if I don't know, if your department also dealing within the climate change. So if you add some component of the climate change, so it will be grateful for us to learn more, like valuable than that suggestions.
Thank you. Just one, maybe, it's easier to talk about it than to work with the placement, but it's also difficult, there are different organizations for the health care of animals. So sometimes it's very difficult to do the arrangement with them too. But anyway, we have an office in the CDC with the sponsor for zoonotic diseases, so we take care of it.
In the Iranian CDC, 4 offices are involved in this. The other is, and for the building, we are working closely in collaboration with facilities in order to governance of ESPL Club. In that project, Ministry of Health, Ministry of Agriculture and Wet, and was the involvement of the human world. Classically, in that project, the one supposes we can give Dr. Rohani, your colleague, they can give the detail of the
इसको बनाना वाजिब है। We're waiting for Ma'am Khazina. So no more questions? If you have any questions from the online participants? No, I think we just have phrases for the presentation. Okay, thank you very much. So with this, we come to an end to our international course on outbreak investigation and control of infectious diseases. I am extremely grateful to our international panel, honorable guests from Pasteur Institute of Iran, and also to our virtual speakers, Dr. Atiyatul Wahab and Dr. Sabah Farooq, for their wonderful and very informative presentations. I'm also very grateful to all the participants who joined us in person and online. Thank you very much. We hope that this training was a great learning and networking opportunity for you all. And inshallah, we'll see you in future important events. Thank you very much. And now I would like to request the panel to kindly present the certificates to the participants. Thank you. Mr. Pawan Kumar. Mr. Murad Alam.